Hello, welcome everybody to the day 2 Restorative Aquaculture OA Mitigation to Maintain Ecosystem Services. We will be giving a couple of minutes for attendees to join. All right, I think we can go ahead and get started and people can trickle in. So welcome everyone. Thank you for joining OA Week 2024. My name is Catherine Brown and I'm one of the coordinators for OA Week. Please note that this session is being recorded. OA Week 2024 is presented by 4 sponsoring organizations. First, there's GOA-ON, the Global Ocean Acidification Observing Network. Second, there is NOAA OAP, the United States National Oceanic and Atmospheric Administration, the Ocean Acidification Program. And third, there is the International Atomic Energy Agency, the Ocean Acidification International Coordination Center. And last but not least, there is IOC UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. Some background on GOA-ON. It was established in 2012 with just a handful of members, but since then it has grown immensely and now has over 1,200 members from 114 countries. GOA-ON also consists of 10 regional hubs, which span across continents and oceanographic regions, and we'll be hearing from most of the different hubs throughout OA Week, so be sure to check out the website to join any of those sessions. If you are not a member yet, you can join GOA-ON today by visiting goa-on.org. I will also drop a link for that in the chat. The first OA Week debuted in 2020 and returned in 2021 when events and conferences were postponed due to COVID-19. Following the successful in-person symposium on the ocean in a high carbon dioxide world in 2022 and another successful OA Week in 2023, GOA-ON brought back OA Week 2024 to maintain momentum around OA research and provide a virtual platform for the ocean acidification community to exchange their latest findings. We are thrilled to have this session on restorative aquaculture with speakers and audience members from all over the world. Just a few logistics before we get started. During the presentation, all participants will be in listen-only mode. You are welcome to type any questions into the question box, which can be found at the bottom of the control panel on the right-hand side of your screen. We will be monitoring incoming questions and will pose them to our speakers during the question and answer section at the end of the presentations. For the discussion, you can also use the raise hand function in the toolbox at the bottom of the screen, and we will call on you to have you ask your question directly at the very end. And with this, I would like to introduce you to your moderator for this session, Veronica Garcia Condo. Ana is currently a research consultant of the Oceanogami Agency in Spain and the Biogenia Agency in Ecuador. Ana, floor is yours.
Thank you very much, Catherine. Good morning, everyone, and thank you for considering me for this role. Today, we're going to experience a very, very exciting session with the ultimate research and advances with regards to multi-trophic aquaculture and restorative aquaculture. Next, we will have our first speaker, which is Marina Martin Sandoval, with the lecture Pathogen-microbiome Early Warnings Aquaculture. Marina Martin Sandoval earned her bachelor's degree in biotechnology with a focus on applied molecular biology from the University of Aberdeen, Scotland, where she completed her final thesis in genomics and proteomics. She pursued her master's degree in Wageningen University in the Netherlands, concentrating on environmental microbiology and studying the microbiome of Indonesian marine lakes. The floor is yours, Marina.
Hi, can you hear me? Thank you.
Yeah.
Thank you so much for the presentation. I will share my screen. Okay. Can you see my screen? Okay, I'll put it in presentation mode first.
Yes.
Let's see. Okay, is it in presentation mode?
Yes.
Okay, thank you
It's
so
good.
much. Thank you, Ana Veronica, for the presentation, and thank you for inviting me to talk in this OA week. My name is Marina, and I'm part of the Applied Microbiology and Biotechnology group in Leitat, that is a technological center based in Spain. And today I'm going to be talking about the pathogen-microbiome early warnings that we find in IMTA labs. This research was made as part of the Astral EU project that received funding from the European Horizon 2020. So let's get first into what's Astral and what was the Astral project. So Astral stands for all sustainable, profitable, and resilient aquaculture. And the main aim of Astral is to develop a new sustainable, profitable, and resilient chain for integrated multi-trophic aquaculture systems. As we know, integrated multi-trophic aquaculture gathers multiple species from different trophic levels into one system. And in this case, this project was composed of several partners. There were 2 IMTA labs, open water in Scotland and Ireland, and 2 land-based in Brazil and South Africa, one a RAS system and one partially recirculated land-based system. There was also a prospective IMTA lab in Argentina. And I believe my colleague Alessio will be talking more about the Astral project. But just to give you more or less a background of where we started and what the aim of the project was. So why is it important to study the early warnings and the microbiome in Astral project in particular, but in aquaculture and IMTA systems in general? So we know that IMTA aims to reduce the impact on ecosystems biodiversity and therefore contribute to a more sustainable and resilient aquaculture, therefore contributing to food security. But on the other hand, all species in this aquaculture system are more susceptible to emerging diseases caused by different pathogens such as bacteria, fungi, viruses, and other parasites. These outbreaks in aquaculture are expected to increase due to climate change, and the open water systems are the most vulnerable ones. So by understanding why the outbreaks of these diseases happen, we can tackle them before they come to us to the threshold level, and we can aim to keep the system healthy. So that's more into the early warning approach, but why is the microbiome important and why do we want to introduce the microbiome approach in this kind of systems? Well, we know that microorganisms play a crucial role in the host health for many species in these IMTA labs. And by understanding the system's microbiome and how they behave and interact, we can stable what a microbiome, we can understand what a stable microbiome would be, and we can keep the system healthy by understanding how the microbiome changes and how they interact with one another. So the way my talk is gonna go today, and the main aim of my talk is to describe the potential presence of pathogens and identify the emerging technologies and monitoring methods for routinary analysis in the IMTA labs to prevent pathogen outbreak. This is a state-of-the-art work done focusing on the IMTA labs in the Astral project, and then using real microbiome data to see how this would look like. So first, I'm going to talk about the early warning signs and symptomatology of the disease and why is this important to understand and know about. Then I'm going to talk about the most problematic pathogens per system and why are outbreaks more likely to happen. And then I'm going to talk about real data from a case study in the Astral project related to the microbiome. So firstly, if we looked into the early warning signs and symptomatology, to understand and characterize an outbreak, we need to take a step back, and we need to understand that it's important to know the symptomatology of a disease for its early characterization. For pathogen detection, it's important to control different factors. So we have different pillars here. We have three, which would be the environment. So this would be how, if the temperature increases, if the oxygen increases, the salinity, for example. We also have host health. If our species are overcrowded or are under stress, or they might have a compromised immune system, they might be more susceptible for an emerging disease. And we also need to take into account the pathogen itself and the presence and absence, and what would be their ideal growth conditions and their host availability. So it is key to, these three pillars are key to early warning characterizations. And by observations of clinical signs and behavioral alterations based on known literature and based on previous IMTA labs or aquaculture systems, we are able to early detect these warning signs. So just as a reminder that we need to take into account the early warning signs and symptomatology for best tool control detection. So we need to once we get to this point, we need to see, okay, what's more important to my system? Do I want to study the environment? Do I want more surveillance on my host? Or do I want to look into the pathogen presence or absence, or quantify this absence? So we looked into tools and methods for either monitoring the environmental IMTA systems, or identification of pathogens and treatment, or the early warning signs of a pathogenic disease by monitoring the stress and behaviors of our species. So here we divided it into molecular tools and biosensors. So for molecular tools, it's more focused on pathogen detection in terms of presence, absence. We proposed PCR for presence, absence, quantitative PCR for the quantification of the pathogen presence, or high throughput sequencing to understand better this, and of course, microbiome analysis. The downside of these molecular tools is that, of course, the equipment is expensive. There's processing of samples involved, which would be limited in some cases, and it's very hard to do in situ without cross-contaminations. On the other hand, we have biosensors that are a cheaper, faster way of detection mechanisms for early warnings, and they also allow for environmental monitoring. For example, here I've mentioned surface plasmon resonance or lateral flow biosensors, which allow for fast detections of pathogens. The downside in biosensors, on the other hand, is that there's a need for specificity and standardization in these biosensors, and that there's a need for research in terms of aquaculture development for biosensors. So to put an example on our literature review and how we came to this conclusion in our IMTA labs, I'm going to talk about the most problematic pathogens described in each of the IMTA systems, and then the proposed tools and sensors in each of the cases. So in open water systems, in our case, we had Atlantic salmon and lampfish, molluscan shellfish, and seaweed, and we identified several main pathogens in these systems. So, for example, we have Aeromonas salmonicida that causes furunculosis disease in Atlantic salmon and lampfish, and it's hard to control, and, for example, causes blisters on the surface. Of course, we have Vibrio species, which we know is very well known in the aquaculture sector. And as we know, is opportunistic and can cause high mortality. To mention a virus, for example, we have the salmonid alpha virus, which causes pancreas disease. And other pathogens, the main one and the one that is mostly talked about is sea lice, that as we know causes skin damage, osmoregulatory failure, and it's very hard to tackle in open water systems. So in this case, we need to focus again on what do we want to monitor? Do we want the environmental monitoring, surveillance, or detection of pathogens first? So in this case, we propose biosensors, such as the wireless sensor network that allows for environmental monitoring in the system, and this would be good for open water systems. The fish operculum, which it's more for the surveillance mechanism for fish, or the lateral flow biosensors that, as I mentioned before, focuses on the detection of pathogens. Again, this need for standardization and specificity in biosensors. For the molecular tools, we propose sequencing, PCR, and qPCR for further research into our open water systems. But we know these are harder to use in open water because it's harder to take samples and process them in situ. If we looked into the land-based systems, we had the land-based partially recirculated system and the RAS biofloc land-based system. Here we had sea urchins, abalone, seaweed, white shrimp, and tilapia. And again, to mention some of the pathogens that we detected in literature review that were more common for our system. For example, apart from Vibrio, the rickettsia-like organisms, which are intracellular prokaryotes causing mass disease, or the abalone herpes virus. We also have in other pathogens, the sabellid, which is a worm that attacks abalone, and although they're not deadly, they affect the growth of the abalone, and they can deform the shells and increase mortality in the future. And we know that the deformed shells or malformations in general can reduce marketability. In this, again, for biosensors, we propose a wireless sensor network and lateral flow. And for molecular tools, would be easier in this case because it's easier to take samples. And we propose sequencing, PCR, and qPCR. In this case, it would be nice, for example, with the Oxford nanopore, because we could get to species level and we can study further the pathogens in this case. So now that we know what bacteria, viruses, and pathogens we have in our system and what are the most likely to occur in our IMTA systems, we need to take into account when outbreaks happen and what outbreaks are more likely to occur when. So for example, in open water systems, we know that they are more susceptible to outbreaks. This is more likely to happen in spring and summer due to the water temperature increases. In land-based, although they are less susceptible to outbreaks, they can have, for example, in the South Africa IMTA lab, high abalone density. And as they recirculate water partially, when the temperature rises in the ocean, they can have harmful algal blooms, which can affect the system. So that's important to take into account too. For example, in the IMTA Brazil system, the rainy season can lead to low salinity, and the high temperature and luminosity can affect the production of the system. So it's not only important to understand how outbreaks happen and what pathogens in our system we have, but also when are the outbreaks more likely to occur and how we can prevent those. For open water, we propose more biosensors because they are easier to work on rather than molecular tools. But on land-based, we propose sequencing, PCR, and qPCR in terms of molecular methods because it's easier to take samples and then process in the lab. And regarding this, I wanted to focus on a land-based system. In this case, I wanted to talk about the microbiome data in South Africa and why it is important to combine this microbiome data with early outbreak characterization. So I'll first give an overview of the system. So this IMTA lab is located in South Africa, and it's called Bafflejack's Abalone. That is a commercial land-based abalone farm in Cape South. And this system works by consistently recirculating 50% of the water through the system. But as we know, harmful algal blooms have a really damaging effect on these systems. And when this happens, they want to be able to increase their recirculation up to 100% to be able to avoid the harmful algal blooms entering the system. So what we did in this study was taking samples, so they consistently recirculate 50% of the water, and then they can increase to 75% long-term, a couple of months, and then 100% recirculation for short-term, so a couple of days. So what we did was taking samples of the 50%, 75%, and 100% recirculation of samples in the system. So to understand the system, we have abalone in tanks, and we have ulva in the system that feeds the abalone, but also it's used as a bioremediation in the system. This water recirculates through the paddleways of the ulva and then goes to the abalone tanks and then recirculates back again. So we took samples of the ulva, outlet, abalone, and inlet water. So the surrounding water, the ulva, and the abalone. And this is divided into clusters that make up a platform, just as an overview of how we took the samples. But what I want to focus on this talk now is how is this microbiome data important for the system and for aquaculture. So through this microbiome data, we were able to characterize our system's microbiome. So we were able to identify what's a stable microbiome in our system in terms of composition, but in terms of how it evolves with time. And not only that, but we were also able to see the interactions between the microbiome between different species in our system. For example, just an overview, this is general plot of the different species in our system and the ones that were significant. And what I want to focus on here is, for example, the genus Glacicola in the ulva samples were really significant, especially in the 50% recirculation, but this lowers down as recirculation increases. But if we look into the water samples that was taken from the system, we see that this genus Glacicola increases at 75 and at 100% recirculation. So this points towards the ulva genus having an effect on the water samples and as recirculation increases. So by joining these pieces of the puzzle, we can understand more how our system works. And if a pathogen were to occur, we would be able to characterize our system's microbiome and understand why is this happening. Another example would be, for example, if we compare an NMDS plot between 50% and 100% recirculation, we can see that at 50% recirculation, the samples are quite gathered together between one another. But as we increase recirculation, especially at 100%, we can see how these samples in terms of microbiome shift and how they are more linked together. So this means that the increase in recirculation had an effect on the microbiome samples. And therefore, with this information, we can determine how our system is behaving and it's being altered by increasing recirculation in this case. So we also can include in this data the environmental factors that shape our microbiome. And by understanding how the microbiome and the environmental data is related, we can further determine what is more likely to happen to our system in the long run. So just to finish, for future emerging technologies, we propose that we should have a combination between molecular tools, biosensors, and microbial data, and that all this data should work towards a predictive modeling for what a good, healthy IMTA system would look like. And then once this threshold would be disturbed, use this as an early warning system for IMTA labs specifically. We also wanted to point out the holobiome host pathogen interaction, so not taking into account only the microbiome, but also transcriptomic, metatranscriptomic, metagenomics to understand further the underlying parts of our IMTA systems and of our microbiome in each of the parts. Thank you everyone for listening. I want to thank the Astral project and all the partners and also all my colleagues in Leitat. And thank you everyone and Ana Veronica for having me. Thank you. I will stop sharing.
Thank you very much, Marina, for your great presentation. Such an interesting topic.
Thank you.
I am very grateful for having please, thank you for the participants for being here. Use the Q&A box to ask the panelists questions. We'll be monitoring incoming questions and we'll post them to our speakers during the questions and answer sections. Let's continue with this wonderful session. Our second speaker is Salaah Eldin from the National Institute of Oceanography and Fisheries in Egypt. The lecture she will be presenting is the importance of ocean acidification mitigation for coastal marginalized communities. Dr. Salaah is an associate professor at the National Institute of Oceanography and Fisheries in Egypt in the Pollution Lab. With a strong background in aquatic physiology and expertise in water pollution, her research focuses on assessing and mitigating the impacts of pollution on aquatic systems, especially emerging pollutants. This is the moment where the Dr. Sally presents her lecture. Carry away, Dr. Sally.
Good afternoon, everyone.
Yes, perfect.
I don't know if you can see my screen.
Yes, it's good.
Good afternoon, everybody. I am Salaah. I am associate professor at the National Institute of Oceanography and Fisheries in Egypt. And today I'm going to talk about the importance of ocean acidification mitigation for coastal marginalized communities. First of all, as we know that ocean acidification is a change in the seawater chemistry caused by the absorption of excess atmospheric carbon dioxide into the ocean. In fact, a high carbon dioxide ocean is confirmed to have a negative effect on marine organisms, including species which the coastal communities' economies depend on. This could be through many ways. It could be a direct way or indirect way. The direct way on commercial species like shellfish and bivalve, and also the indirect impact through food web and the interaction and loss of marine habitats such as coral reefs. Why we care about ocean acidification and how this is going to affect the coastal communities. The coastal communities, especially marginalized group, rely heavily on the marine resources for food and income, and also it's more like a cultural practice. So ocean acidification, it's have been proved that can impact the marine biodiversity fisheries and ecosystem services, making these communities particularly vulnerable. So when it comes to the ocean acidification impact and how this is going to impact these communities, we have either a impact on marine ecosystems through disruption of food web by a massive mortality or maybe a disappearance of certain species like a bivalve or maybe the shellfish. Also, the biodiversity will be denaturated because the absence of certain species will also affect the higher level of the trophic chain. In the second impact, here we don't to mention only three significant impacts. The second one is the impact on fisheries. The small-scale fisheries, these fisheries are a primary source of food and income for coastal communities. And this is especially vulnerable to change in fish stock and abundance, especially in low-income countries. When it comes to small-scale fisheries, we all know that fish is a very important source of animal protein. And in some countries, especially in low-income countries, it's the only affordable source of protein. So here we're talking about a very significant issue about food security. Also economic loss, because decreased fisheries yield can lead to economic hardship, especially for communities without alternative livelihood. Here we can say, I want this to move. Okay. Here we can see that a small-scale fisheries, this is a chart of or infogram about numbers. Here we can say that 37 million tons of small fisheries have been captured lately. This is according to the FAO statistics, and only about 429 million people have been depend at least partially or engaged in small-scale fisheries. I want to focus here on the role of women, because about 45 million women have been participated in small-scale fisheries, which give a significant opportunity for empowering women. So when it comes to the third impact is the impact on ecosystem services, because coral reef, shellfish bed, and seagrass is an important habitat for several or many species in the aquatic ecosystem. So if there is any denaturation happened, it will affect, of course, the whole system. Here we can say the biochemical or the biological indices that have been cited as affected by the ocean acidification and ocean warming, and the interaction of these two effects on invertebrate and fish. We can see that although the ocean warming is having a tremendous effect on every level, also ocean acidification had a significant effect on these levels, even more than water or ocean warming in fish and invertebrate. This is shed lights or highlights the impact, the profound impact of ocean acidification on fish health and aquatic animal health. So it's difficult to predict how the ocean acidification is going to affect a certain ecosystem, because it's a vary depending on the species, the habitat, the region, or even other variables. So the vulnerability of marginalized coastal communities, this is going to be specific. How this is going to be affected often include these communities mainly have low-income families, indigenous group, and women in coastal region who depend directly on the marine resources. So lake access of resource to technology for adapting the environmental challenge is another feature that really highlight the suffering is going to be in this area. Also, climate change in this area is more profound effect, because it's subjected to many factors such as sea level rise and increasing the storm intensity. So this all going to be along with the effect of ocean acidification. Also, the limited access to education and healthcare, especially when migration happens, if these ecosystems suffer from changes, make this adaptation and resilience building effort even more difficult. Also, the social and economic consequences, because fisheries and fishing in this area, especially in low-income area, it's more like a cultural. It's not like it's more like a habit. So health and nutrition is another aspect of the problem, because declining fish stock is going to have a nutritional effect. Also a cultural impact, because all the village or all the city is meeting at the shore waiting for fish, start to trade fish and start to harvest. So also livelihood losses, because declining fisheries will affect the fish stock in this area, and increase poverty, because it could be a very significant source of income for these nations. As I mentioned before, women has a significant part in the small scale fisheries and in this sector. So in this area of marginal and coastal communities, women have a chance to make their living or to have a stable income. So as the 25% of reef fisheries harvested are mostly by women and children collecting shellfish and other species inside that area. So women are often more involved in fish processing and selling. So reduced fish stock will affect this opportunity of empowering women in this area. A specific negative impact of ocean acidification on coastal communities, reduction of food availability and food security, as I've mentioned, at this community is going to be on many levels, like community or local or even regional level. Also changes of employment opportunity. Many of these people will lose jobs if the fish stock will affect due to the ocean acidification in this area. These are very vulnerable area, and especially in low-income areas. Decline in marine recreation opportunities will affect the number of tourism, the income of the tourism activity. Also, as I've mentioned, that it's more lacking cultural and sociological habit. So loss of cultural value related to maritime and fisheries tradition, to resource management practice, and to spiritual well-being, particularly in cases of indigenous communities. Into the social and cultural changes result from decline in fisheries economy and manifested through increased conflict, migration, and unemployment, and alter the gender relationship, as the traditional role of men and women will change indeed. So also the disappearing of the small and more isolated coastal fishing communities as out-migration reduce the community size and also the service that will be provided. Below the critical mass needed to provide a variable support like schools, health clinic, and transport links. So changes in trade laws and the related value chains will also occur, because there will be spots or maybe area that lake of fish or certain economic value of fish. So this also will affect the trade laws. The loss of secondary economy in such areas such as boat building and all the logistics that comes with this activity, port services, and sale of supplies. So loss of ecosystem services such as coastal protection, as reef denaturate, this will affect the whole process. So who faces the high risk? This is the risk, and we have the coastal area or the coastal marginalized communities. There are part of the world that is can sustain or can mitigate, and the other part is more vulnerable and will affect more severely. So community with high resilience to ocean acidification is the coastal communities that depend on aquaculture production system, which have a significant control over a key factors of the water quality, like and food supply and survival rate. So these communities will have more resilience. Also, the communities that depend on integrated aquaculture, where different species like fish, shellfish, and seaweed are farmed together, providing better use for available resources and reduces stress on individual species. This also going to be a added value for this community. A community that counts on fish cages as also can be removed to alternative site to mitigate this ocean acidification ramification. This is going to be a solution to reduce this risk. Also, the fishing communities with robust fisheries and aquaculture governance regimes and management practice is going to be more resilient to these changes. On the other side, the communities that less resilience to ocean acidification is poor coastal communities that rely heavily on harvesting or capturing species that are sensitive to ocean acidification and that have limited alternative economic opportunities are likely to be most vulnerable to the negative effects of the ocean acidification. On the responses side to mitigate ocean acidification, we have a quick notes on points that could really help this challenge, like empowering coastal communities, like mainstream ocean acidification into climate change policies, plans, and investment strategies at communities and nation levels. Also raising knowledge and awareness about ocean acidification impact and responses. Maybe building a resilience and sustainable fisheries and aquaculture will help a lot in this issue. Also accessing climate finance to rebuild or build carbon blue sink, such as mangrove forest, will help. Also fostering public and private investment in social and economic environmental capital. This will help the cause. On the other side, empowering fisheries by improving management of coral reef fisheries and sustainable fisheries to adapt innovative to add local value through fish processing and using polyvalent fishing vessels. This going to help on this level. Empowering mariculture, supporting seed availability for closed cycle mariculture, and also selecting more resilient species and culture systems. Using selective breeding, maybe like using a DNA barcoding or maybe any innovative techniques that could help in this point. Also developing monitoring and early warning system could help to detect any fluctuation or change in the water. Managing coastal zone and conservation of carbon sink also is going to be an added value for this issue. A recommendation, it's a few quick tips, and this issue is a way beyond. So I here try to gather the most significant recommendation, like reinforce the climate change mitigation agenda or raising awareness and knowledge at all levels. And this point is very important for the public. Also mainstream ocean acidification into national and regional and even global policies, and improve ecosystem and community resilience. Also, the financial action is very important to support the ecosystem and the community resilience. We need also to support and extend initiatives to monitor ocean acidification and link it stressors, and there is a huge programs really now working on this point. Thank you.
Thank you very much, Dr. Sally, for your great presentation. Thank you. That was very enlightening. Next, we will have our third speaker, which is Sebastian Gatica Montero from HuiroRegenerativo. The lecture is Regenerative Whiiro. Sebastian Gatica Montero is a master and PhD from UCL in London and a commercial engineer from the PUC in Chile. He's a social entrepreneur, academic, and director of Collab UC. Please, Sebastian, go ahead with your presentation.
Okay, thank you very much, Ana Veronica. Can you hear me well? It's all right?
Perfect, perfect.
Okay, let me just share the presentation. So here we are. Yeah, so thank you, everyone. Yes, I'm a co-founder of Whiiro. So in that position, I will share with you some of our perspective and lessons learned for our company. I'm not a marine biologist, but I've been involved with seaweed for a couple of years now. I'm involved in innovation and regeneration. So a part of the regenerative paradigm that is emerging elsewhere and also in Chile. So we titled this presentation as From Sustainability to Regeneration. So it's a journey towards restoration of local communities and coastal communities. So yes, so what we're facing today, we just want to frame this and how we built up and we create this company and a sense of urgency that we're facing this poly crisis and also the systemic nature of the crisis that we are facing today. Also, we have tried to learn the full process of how entities and institutions, multilateral agencies, and so have been discussing issues like planetary boundaries, sustainable development, biodiversity crisis, and so on and so forth, until the point that where we are trying to face resilience, we're trying to understand sustainability and beyond that, how to actually, this panel is also about restoration. So we try to merge all this perspective to try to bring our own solution more from innovation and regeneration towards seaweed and coastal communities. So this sense of urgency is also framing that the perspective that we just passed kind of the overshoot days, and we as Chilean have been seeing like the last decade how we are moving towards a country which actually damage and we go far beyond our capacity and Earth capacity to regenerate itself, I would say. So yes, in that sense of urgency, we are trying to understand what this Bill Reed and the people that work in Regenerative group trying to frame and to lead us towards this illustration where we position like our development model, I would say, and how we can integrate this human consciousness, moving towards the degenerative paradigm, I would say, towards a restorative, regenerative, and regenerative development. This is how we try to understand and think that we are pushing that and try to bring this invitation in a way towards this new paradigm towards the seaweed industry somehow. So seaweed as cultivation as a response to these multiple challenges. So beyond all the positive impact or the opportunities that we might see in the seaweed industry in Chile, we try to to embrace that. So just first understand how we are positioned globally in terms of seaweed. So we are a country that are quite a large exporter of seaweed, but mainly our exports are just behind countries that actually farm all their seaweeds. Like we as Chilean, we export a lot of tons, but only 4% of them comes from farming. So it's just extracting them from natural seabed without really a sustainability approach or practice and so on and so forth. So we do understand that we need to move beyond that and also to understand carbon neutrality on the full model or our impacts or a more systemic perspective on that. We also understand that 80,000 people live as harvesters. So there's a full coastal communities involved somehow, but really it's even near to offer them sustainable livelihood. They're just surviving somehow the full ecosystem and operating from extracting towards these intermediaries that actually sell the algae elsewhere with a declining of our prices, declining our position in the global market. So it's kind of a crisis within the sector and also within the ecosystem that are there. So we need to advance towards a new model. So in that perspective, we create Whiiro, and we actually call it, our company name is Aquaculture. So we are trying to explain local communities and local farmers that they will be as the agriculturalists, as farmers, but in the sea, in the coastal community. So we're trying to make things simple. So we're trying to bring all this regenerative perspective really in a simple technologies and even simple language for them to try to understand and to try to move towards the generative paradigm towards a regenerative paradigm. So through the use of seaweed cultivation as sustainable management naturals bed, aim to achieve this environmental, sociocultural, and economic regeneration of marine ecosystem and stakeholders within the system. So this is kind of our purpose. We have the 3 pillars in regeneration, like environmentally, socially, and economic. And we have been coming up with 2 main models. It's kind of context specific, of course. We're trying to understand which are the possibilities of this kind of to hack the system somehow, to try to work with local actors, try to see how they're like a paint in a more like an innovation language, how we can add some value to it and help the full ecosystem for aquaculture ecosystem in Chile, which is quite large. We do have like a salmon industry, we have mussel industry, and they're kind of large globally. So we're trying to use this force, their need to actually to build and to transform the full coastal communities for the good. So we have this collaborative and circular model with the actual aquaculture industry, and also a more like a community model with a regenerative approach to it. So the first, we are trying to understand the aquaculture's kind of needs in terms of this use concession that they have and the infrastructure available, also the plane available in terms of their productive models. So we work with local communities that are like a side of these sites of aquaculture, so they're living there, they're close by, they're looking at aquaculture as a possibility. We try to involve them, to work with them, and to somehow involving them in seaweed farming as a space to learning new things. They're moving towards reduction, the impacts, and working towards regeneration. So this is the very first model, and we work really close by the industry and try to work with them and their local community that always see them as a conflict. We're trying to put in a win-win situation. So in Chile, as you might know, there are as I explained before, salmon concessions elsewhere, like a lot of them, and at least 900 of them, like a large size in the sea that are available, some of them with infrastructure, but they need to do something on them. So we are trying to, what if we use like a part of these concessions to involve communities to learn a full process of seaweed farming? So they're paid somehow by the salmon industry to get involved. We are moving somehow to this multi-trophic prototyping and see how they can, or even like a semi-integrated, not maybe in the same concession. So we are moving to that experimentation as some of the colleagues just present. So if we took some of them, at least 200 of them, we can somehow sequester or capture, let's say, capture 100,000 tons algae, and also 2,000 fishermen could be somehow transformed towards this regenerative industry rather than the extractive industry. So we are seeing that as a possibility. And the second model is just directly driven for community and with communities, I would say. And we're working with them in terms because they do have the possibility of farming their own a site that they have. But it's kind of the issue of governance, how to get them involved and to attract them towards to do this shift, to involve. So we are working with community more with women, we are involving them, and we provide all the initial investment they need in order to set the seaweed farm. We acquire somehow the crop and the knowledge technology, and they make it available that is paid. So we pay with the labor, and also we pay at the end for the seaweed. So community return investment by widow with the algae somehow. They return their investment with the algae, they pay them us with the algae, and this we build this active interactive model that is now performing somehow as a tool for education and transformation of the food industry. So this is some of the examples that we're working. We try to use a really simplified technology. We do have some central hatcheries, which are much more developed than this that you can see in the picture. But these are kind of prototypes that we are using, kind of low tech, but easy for them to understand the principle behind the hatchery, and they can get involved in the full process. So they're kind of capable of doing and replicating them afterwards. And also as a place throughout the fjords in Chile, which are spread all over the south of Chile. So we are working with them, with mainly women, I'd say. Also, we use discard material from different, from the aquaculture sector. And we are kind of making this progress, and so far, so good, I would say, in terms of the full process after farming the algae with them. So we are working in all different dots, like this red dots are our farm somehow with local communities, working with the industry. As my of the logo, Aqua Chile, for instance, is the second largest salmon company in the world after Mowi. We are working now with Mowi as well, which is a Norwegian company. So we're working with pretty much all the companies like a salmon industry and involving them. So as a tool for transformation towards this regenerative model. We understand and our value proposition comes mainly through collaboration. So we have created a full model of Huiro pretty much as an ecosystem. So it's a newly born ecosystem in terms of farm seaweed. As I mentioned, we are kind of pioneer in this world of building this collaborative model. I mean, there were people farming before us, but understanding this as an ecosystem that comes, the value proposition comes from collaboration, I think that is far new. Last year, we won actually the Innovation Award in Chile for that. And we have kind of tried to set up the full value chain, the full process, and trying to run smoothly that. So this is part of our new hatchery, the more central hatchery, working with companies, for instance, Ortimare, that most of you might know them, that are based in Holland. We are partnered up with them, trying to bring all their technology. We are moving to a sea farm. Our cultivation so far as getting all the protocols has been working really good. So we are working and moving towards monitoring our crops. Like we're working with EDF, now with the Nature Conservancy, so Environmental Defense Fund. So different entities that are involved in this regenerative paradigm and trying to bring that measurement, all our scientists behind. At the end, towards this harvest and kind of value creation that we are just involved already in biostimulants, but moving towards like a cosmetic industry, more feed. We're working with Nestle, with different companies to try to make the most of hopefully protein, working with FAO as well. So we're trying to push this like towards an opportunity to scale up and to transform the full ecosystem through regenerative farming. So this is our full value chain. Sorry for the Spanish there. So this is our latest kind of harvest. You can see the pictures above. And we are measuring all the carbon, the phosphorus, biodiversity, nitrogen. We have been measured for 3 years in every farm. So we have measured for all of that. We know that we are capturing somehow. We need to discuss, and this is where we are involved in how to actually sequester the carbon at the end. So biostimulant might be an alternative, but not so. I think that the ratio is not that high, so we are still discussing which might be a position or a possibility to actually sequester the carbon at the end for more than 100 years or so. So impact today, we have more than 10 hectares farm, like the CEO that captures, like nitrogen was mentioned, the families involved, and the people that we have trained already as seaweed farmer. It's something new in Chile that we're proud of it, and the value and the force come from communities, which might be a slightly different approach that other seaweed projects elsewhere in the world, which is this mainly through kind of medium or small side, like farms, involving the communities and with them to scale the full movement as we aspire to move from sustainability to regeneration. That's it. Thank you.
Thank you very much, Sebastian. That was amazing and inspiring, as all the research our panelists have shared with us today during this Go On workshop. That was amazing. Thank you for sharing with us. Okay,
Thank you.
next. Thank you. Next, we have our next panelist, Dr. Alessio Gomerio from Astral Project. His lecture is called Integrated Multi-Trophic Aquaculture, the New Value Chain in the Atlantic Ocean. Dr. Alessio Gomerio is senior scientist at NORCE and scientific coordinator of the North Atlantic Center. He is specialized in chemical quantification and characterization of polymers in environmental matrices and their associated biological effects on model organisms. He is a project leading research initiatives toward the method development to state micro and nanoplastic occurrence and composition in marine freshwater and terrestrial environments. Go ahead, Dr. Alessio.
Thank you, Anna Veronica, and thank you very much also for inviting me to contribute in this very interesting webinar, and everyone to attending to it. So now I will introduce a little bit what the Astral project is, and providing you with the results associated to this specific project. So what is Astral? Astral is a European Commission funded a project of 8 million euros in 4 years, and has been like integrating and complementing a consortium of a multidisciplinary and multi-sectorial capability with partners across 3 different continents. The project per se has been helping with the implementation of the BELEM statement, and also the collaboration across European Commission, Brazil and South Africa in terms of agreement for scientific and technological collaboration. I believe that now we know all about what an integrated multi-trophic aquaculture system here. So it's a co-cultivation of a feed species together with extractive species in order to compensate and reduce at maximum the fingerprint, the ecological fingerprint associated with this type of production. IMTA's, multi-trophic aquaculture system, may be completely open, so cages suspended in the water, or could be completely open or completely closed, like semi or total recirculating system. In Astral, we have been like keeping a specific eye on the zero waste, try to figure out how to reduce at best the possible ecological fingerprint associated to the cultivation of species, and the secondary also to identify the best condition for the cultivation and identify new species of economical relevance in order to create business models and application for boost the local and the general economy across the different partners in the project. In this project, we have been targeting 5 different case studies, as my colleague Marina has also introduced in the previous presentation. One is located in Ireland, one in South Africa, Scotland, Brazil, and Argentina, where Argentina is a proposition area for the cultivation of different species. This type of case studies has been analyzed not only for the possible perspective increase of the production, but also for the identification of new species, and also with an eye on the possible species welfare, so for the health management and also for the biosecurity and the potential environmental impact associated to the production. At glance, the first production in the first IMTA lab, you may see here a picture, was somehow representing a different type of production. I'm not going too much in detail about the different interaction, but in the first IMTA, you may see that we have a fish, fed species identified on salmon, a fish, and then a couple of extractive species in order to get rid of and uptake most of the nutrients that are not used or released in the environment by the feed species. Plus, there was like a plan to develop new species and try to test their production, and if it was feasible, to cultivate them, and what was the interaction with the pre-existing species. And this was like a purple sea urchin, Paracentrotus lividus. Another case was the the completely recirculating system located in Brazil. In this specific RAS system, there was applied a biofloc technology which means like a combination, a mixture of organisms like protozoa, zooplankton, microalgae, and heterotrophic bacteria, which have been like serving as a sequester of ammoniacal nitrogen, phosphorus, nitrogen, and carbon, but also as a food source for other type of species. In general, the species that have been like cultivated in the Brazilian IMTA were like a white shrimp, tilapia, a fish, a native oyster, seaweed, and sea asparagus. Different type of parameters have been like tested for checking the efficiency of this kind of system. It is important to observe that in this specific case, since it's a completely closed system, there is no water renovation, so no use of waste of water in this kind of type of production. And this type of production is also classified with high productivity, way more than other type of production. We also have like addressed some production in South African IMTA. This is a land-based pump assure system, which is partially recirculating water. So some of the water after use is discharged again, and the new collection is taken during the production. Again, for this type of production, we identified at the beginning feed species, so abalone, and extractive species like algae, uva lanceolata. Together with that, there was also a new tentative species combination in order to boost the production, new extractive species, and also new other species identified with the sea urchin as a feed probiotic for juvenile abalone, attempting to short-circuit the production inside the IMTA system. The Scottish IMTA again identified like some inorganic extractive species, algae, and also some organic and inorganic extractive species identified with the king scallop and native oyster. In this IMTA, again, there was also the possibility to experience and experiment the introduction of new species, which in this specific case is the edible sea urchin and the Palmaria palmata. In particular, for this IMTA, we should record that within the project, there was a successful cultivation of 5 new species, 3 bivalves and 2 seaweeds. And then there was also like the possibility to experience and optimize the cat cultivation methodologies, but also identify like a wrong turn or area where no further research should be like addressed. So the proof of concept and lesson learned, which is centered across the echinoderms, which at the moment, according to the results of the project, is not suitable for cultivation, and also for the red seaweed Palmara palmata, where we actually are on an embryonal stage where there are like some advances in the seeding process. We also have like a prospect IMTA, as it was like mentioned before, located in Argentina, in the Patagonia area, in the Terra del Fuego, where like there is like some plan to start the cultivation of blue mussels, red sea urchin, and also install a land-based open system, which will be targeting a couple of fish species and also an inorganic extractive species. There has been like a different type of integration across the different case studies, but at the end, what the project was aiming at releasing was also technical guidelines, somehow focusing not only on the economical aspects associated to the introduction, the interaction of new species with other species, so the so-called development of a business model associated to the production, but there was also best practice and documentation about the environmental impact associated with the production, and suggestion about how to implement the circularity associated to the different type of production. Again, one of the main focus was like to reduce the waste associated to the such type of production, but it was also important, and it was also focused to run the circular economy assessment in order to identify gray area and area where implementation can be put in place. Here is like a very schematic and very summarizing results associated to the life cycle assessment. I will not ask you to go more in detail about this, but you may see that there has been a lot of positive evaluation associated with the integration of multi-species across a single area, with interaction and integration in order to reduce the ecological fingerprint of this type of products. But details can be connected while extracting the results of the project. So I basically invite you also to go there and then collect all the different reports that are associated to this. Together with the life cycle assessment, the project per se has put also a magnifying lens about the socio-economical aspects of the multi-trophic aquaculture system. So the different actions associated to this type of investigation was also collect a state-of-the-art analysis of aquaculture, and then try to identify mesoeconomic approach based on the comprehensive bibliography review, and then develop a business model associated with the case studies, and attempting to create some methodological criteria for the evaluation of the economy associated to the production and the launch of new species using SWOT and PESTEL diagnostic methods. In general, what has been the main results associated to the analysis is there are some economic dynamism to the coastal area, adding a new species. There is an higher increased net present value, which is going by without saying, because we are somehow increasing the biodiversity of the product. In terms of market vision, there is a limited exploration so far about the potential market and a lack of customer understanding about this type of production. There is also the need for a producer of development a market strategy considering these emerging markets. I'm presenting also this methodological approachment about the assessing of the circularity in general, with that we applied to the different IMTAs, starting to monitor and studying the innovation about the feed rate, feeding technology, the contribution about the extractive species and the capability of bioremediate the emission, so collect the escape of phosphorus, nitrogen, and carbon from the feeding species, and then try to identify best practices associated to the open system, and try to understand the circularity in this specific area. So we identified, while trying to analyze the circularity, different type of indicators. So they made the bioremediation, the feeding technology, their use of water, their use of energy, and also the use of infrastructure material, and then try to identify this into specific circularity metrics, like the resource, the efficiency of the use of the resource, the degree of recirculation, the utility, and of course, very important is also the effects associated to the environment. In general, all the different IMTA lab behaved well, with some more or some less efficiency. Again, here we have basically described all the different type of parameter that has gained a little bit of a value in terms of circularity across the different IMTAs. So in general, about the circularity, we did our effort in Astral, but we also need to record that there is a lack of standards to measure the circularity in aquaculture production. We hope through this project to have provided some useful consideration and useful feedback in order to create a better standardized way to extract and analyze the circularity. Of course, because of the type of setup and the type of case studies we have been analyzing, there is a larger variability and a complexity if you compare open, enclosed, or semi-closed system. And we should also, discriminate, or at least mention that when you're thinking about recycling and circularity, is not only nutrients we are thinking about, but it is also the better use of the energy and the natural resources. Astral has been also like a good example and a good exercise to investigate also the environmental impact associated to this type of production, but also how environmental stressors may influence the production. And this is also something that is very close to my heart, my scientific heart, because as it was like mentioned, I am a specialist in microplastic analysis. Here we have been identifying a different area of investigation. Some have been also already presented by my colleague Marina. But we also try to link what could be the effects in relation to the climate change, in relation of the occurrence and the release of harmful algal blooms, pathogens, and microplastic. The project per se has identified the potential risk and link them with the climatic risk, and also some suggestion how to mitigate this risk have been also provided to the proceedings and to the different deliverables associated to the project. In terms of results, these are presented in terms of open ocean system, land-based, partially and fully recirculated. Again, I'm not presenting a single results step by step, but I will invite you again to enter into the website of the project and download all the different information that are open access available through the project. This has been also disseminated in a different type of conference and in different type of platform. So in general, in terms of monitoring this type of infrastructure, of course, there are some general recommendations that we should maintain the well quality of, and we should control the quality of the environmental measurements in the long term in order to be able to monitor if there is a depletion of the quality of the area in the nearby of the aquaculture site, also in relation to the climate change. The assessment of the harmful algal bloom show a very complex interaction with the biological, physical, and chemical factors. And of course, our attempt to generalize a little bit the type of behavior failed, because there is also a regional environmental knowledge which is of paramount importance when you are assessing this kind of effect. The project per se, Astral, together with offline type of analysis, has been also boosting a lot surveillance strategies based on a biomolecular tool, biosensor, and sensor in general. So together with what is carried out in the lab, we also tried to automatize remote sensing and simplify somehow the measurements. So there is also recommendation about how to at least pave the way for remote surveillance of some of the parameters that we have been analyzing. In terms of specifically on the microplastic pollution, something that we need to report also here, that there is a general uncertainty about the knowledge associated to this type of pollutants of emerging concern. The knowledge is at the moment sitting on the researchers, but when you're talking about stakeholders and general general people, still there is not really a clear idea how this kind of chemicals of emerging concern are interacting in the aquaculture system and with all the derived consequences. So Astral is also, again, because of this high technology developed platform, also suggesting artificial intelligence data analytics platform. So something that is collecting, extracting all the raw data from biosensors, spectrophotometers, remote sensing system, vision sensor, in order to then be integrated and then reported in the appropriate way. This is something that, in our opinion, may be represented the future of the monitoring of the quality of IMTAs. So it is something that we also wanted to mention here. Again, this has been also collected on a specific deliverable in the Astral project. And again, I invite you to have a look and then get inspired about the output of this specific research investigation. Astral is not only science, is not only production, is not only economy and business, has been also a good platform to integrate and exchange information with the normal publicum, with also collaboration with the specific stakeholders, where like workshop, webinar, and other type of dissemination in the school, questionnaire, and other type of tool have been helping a lot to exchange information, not only across the scientific community, but also with the normal people that are interested about aquaculture and how aquaculture is evolving. So, thank you very much for having attended this presentation. These are the contact information about the project coordinator, my colleague Eliza Ravagnan. Thank you very much.
Thank you very much, Dr. Garcia, for your wonderful presentation. Such an enlightenment information and such a wonderful research. And thank you to all the panelists and speakers that today have shared such a wonderful work you're leading towards restorative aquaculture. Any question, please fill the section in the chat so we will further answer your questions and get in touch with the panelists that today have presented the topics of their research. And this has been the last presentation of today's workshop focused on restorative marine aquaculture. If there's another mention.
Hi, thank you, Anna. If there aren't any questions, or if you would like to chat offline with any of the participants, that is completely okay. Thank you so much for the presentations today. I am going to share my screen for just a moment. Okay, thank you again to all of the organizers, the moderator, and the speakers for a great session. We would also like to thank you, the audience, for joining us for OA Week 2024. Please don't forget to check the website and to register for any other sessions that might interest you. If you would like to stay up to date with the Goa On community, also please consider signing up as a Goa On member. We sent that link in the chat just at the beginning of the session. There's also an open call for the Goa On Executive Council. So if you're interested in more information or to nominate yourself or someone else, please just scan the QR code on the screen. Thank you all. We will see you hopefully at another session, but if not, we'll see you next time.